My name is Pourya Naderi, and I'm excited to present my research to the GLBIO community today. So the fundamental question of my talk today is to understand how well does a model disease mechanism match the actual human disease? Therapeutic target discovery in complex diseases, including Alzheimer's, can benefit from answering this question to close the gap between human patients and model systems such as cellular models or mouse models. In the case of Alzheimer's disease, there are currently more than 6 million people in the United States alone with the disease, and there are multiple models available to study disease, including iPSC models and transgenic mouse models that recapitulate different aspects of Alzheimer's disease pathology, such as neurofibrillary tangles and amyloid beta plaques. Treatments of Alzheimer's disease have been successful in preclinical trials and models, but there has been limited success in human clinical trials. This immediately leads to the conclusion that action in models does not equal to action in human brains. So when we evaluate how good a model is, the proxy is phenotype, how well specific disease pathology is recapitulated in a cellular model. When proxy is phenotype, we don't know if the model system and humans recruit the same set of genes and pathways to drive the disease pathogenesis. Consider this hypothetical example where the same pathogenic cascade recruits different gene expression profiles in a cellular, in a dish, and in a human brain. What if we had a simplification to represent the activity of groups of genes as one number? We know that there is inherent noise and subtle changes in the gene expression that might not be necessarily capturable. So if we could look at pathways as units of activity, and if we could determine directionality of functions in terms of dysregulation, whether a function is upregulated in the disease or downregulated, then we would have a basis to compare pathways in a cellular model or a mouse model versus human brain. So today I will present integrated quantitative pathway analysis, which is a framework to unify representation by quantifying pathway. Essentially, IQPA will determine the appropriate pathway in an appropriate model to target as therapeutic candidate. And we're essentially making function portable by finding conserved pathogenic pathways that have concordant dysregulation in a variety of brain regions and model systems. Before I start to explain how IQPA finds high potential candidates for Alzheimer's disease, I want to spend a few minutes to talk about how can we quantify the activity of pathways in IQPA. So the main inputs of IQPA is first a gene expression data set, preferably that has disease and control subjects in it, and a database of annotated pathways. So for every single pathway in an annotated database, IQPA masks the expression of genes that belongs to that certain pathways and aggregate them into single numbers that represents the activity of pathways. I will not go through the technical details of IQPA much. You can read the details in the papers that are cited here, but I want you to know that this approach is a rank-based approach that simply, to put it in simple terms, it averages the rank of genes in a descending order. So genes with higher expression get a higher rank value. And having this system now, we can address a series of questions. We can take different brain regions and cellular models. We can treat pathway activity profiles the same way we treat gene expression profiles and do quantitative approaches, linear models, compare them between cases and controls, and find similar pathways with similar dysregulation between model systems and human brains. So one of these questions immediately is to understand what are the common pathogenic functions in different brain regions. We know that Alzheimer's affects different brain regions in a different order and probably different functions. So in this case, I used two different brain regions from cohorts of Alzheimer's disease one from temporal cortex, from Mayo Clinic cohort, and parahippocampal gyrus from Mount Sinai Brain Bank. And I determined the dysregulated pathways between 80 subjects and control. So immediately we see that there are, out of about 1,200 pathways that we analyzed, there are 277 pathways that have dysregulation and in both brain regions. And interestingly, these 277 pathways almost always have the same direction of dysregulation. So the pathways are either significantly upregulated in both data sets or significantly downregulated in both data sets. So here's a snapshot of the heat map of the pathway activity profiles of these 277 pathways. I will not go through the detail of how many of these pathways are actually known as key pathways of Alzheimer's disease. But I want to explain that by using the dysregulation metric of pathways between cases and controls across data set, we can actually establish a measure of similarity. So we can calculate the T-scores and plot them out for different brain regions. So in this case, you see each dot represents an annotated pathway. X-axis is the dysregulation of the pathway in Mayo cohort. Y-axis is the dysregulation of the pathway in Mount Sinai cohort. And we see a strong correlation of 0.84 between the two brain regions. I'm sure that by this point, some of you are asking, so why couldn't you do this just by gene base? And we show that actually, if you do the same exact analysis based on gene dysregulation, basically T-scores of gene dysregulation, you get a substantially lower correlation. Now, we can extend this analysis, correlation analysis, to include multiple brain regions and multiple cellular models. So in collaboration with our experimental colleagues at Mass General Hospital, we analyzed multiple cellular models that are based on different mutations in genes that confer different pathology of Alzheimer's disease. I will not go through this table. I know it's a bit overwhelming, but the point of this table is to show that each one of the models can be compared to different types of controls, and we can look at different aspects of pathology. So now, if I calculate the dysregulation statistics for each one of the models and add a couple more brain regions, we can immediately establish a similarity map that you can see on the right between different brain regions and model systems. So for example, here, Rose map represents dorsolateral prefrontal cortex, Mayo, and Mount Sinai are already introduced. But what I hope that you can appreciate is that one of the model systems that recapitulates amyloid beta 42 versus 40 ratio pathology, in fact, positively and significantly correlates with different brain regions. And upon unsupervised clustering, this model actually groups with human brain regions rather than other cellular models. Now that we've established a similarity measure between models and human brains, the question is, what are the pathways that are driving this similarity? And if we can action them. So in fact, when we consider this cellular model and the three brain regions, we found 21 pathways that had significant dysregulation between cases and controls. And we decided to start testing these pathways to see if they have therapeutic value. Before I go to the therapeutic results, I want to show you this plot, and I hope that you can appreciate the genes that belong to the P38 MAPK pathway, the first pathway that we tested. In some of the databases, in some of the data sets, the genes that belong to P38 MAPK pathway were differentially expressed, but in some of the other cases, none of the genes were differentially expressed. Whereas IQPA detected the pathway as significantly dysregulated. So the take home from this is that IQPA can discover pathway dysregulation even when the genes that belong to that pathway are not necessarily differentially expressed. So we decided to test this top pathway which has been known to have pathogenic roles in Alzheimer's disease. And we hypothesize if we target the same pathway in the cellular model, because it is similar to human brains, we should see amelioration of Alzheimer's disease pathology. And indeed, first we confirmed actually that the P38 MAPK is activated by Western blot assays in the amyloid beta 42 high model. Second, by targeting using a known inhibitor, we saw a significant reduction of phosphorylated tau pathology in the amyloid beta 42 high model, as well as reduced cell death marked by cleaved caspase. So in conclusion, I hope that I have convinced you that pathways provide powerful targets for drug intervention, and conserved pathways can be detected across different brain regions, different cellular models. We can find these conserved pathways and target them, then essentially making function portable. And this pipeline, in this case, has resulted in a potent therapeutic target that can reduce tau phosphorylation in cellular models. I would like to acknowledge our colleagues at Beth Israel Deaconess Medical Center and Mass General Hospital. Special shout out to Sang-Soo Kwok, who co-led this project with me, and I will be happy to take your questions.
We have time for one question. Yes?
Great talk. We've also found in our group that pathway level measurements are more reproducible than analyte level. So I thought that was a really clever way of leveraging that property. So I guess what I wanted to comment that for IQPA, so it's called, there are other methods out there that kind of do what you are doing, wherein you consolidate individual analyte measurements into pathway level. I've used GSVA for that in the past, but what I was most interested in is that there's network level approaches that represent pathways as a collection of nodes and edges. And the advantages of those is that they can actually quantify sub-processes within pathways. Because I think maybe the drawback of reducing down to a single pathway reading is that you can miss subtle changes in directionality. Do you have any experience with trying that out, or do you think that these pathways are mostly just unidirectional?
Yeah, so I'm just going to go back to the slide. It's a great question, and you open up a can of worms. So, sorry if I can get here. All right. So, of course, there are many approaches to basically have a pathway representation. Each one of them have their own advantage and disadvantages. So maybe you get higher precision in nonlinear models, but you don't get a realistic value. So this system, one of the advantages is that if you have a higher activity values for a pathway, you can infer that genes that belong to this pathway tend to have higher expression in certain conditions, similar to GSVA. But unlike gene set enrichment analysis method, this one doesn't depend on a null distribution. So you can, and that's, I think, one of the cases that we can make the pathways portable between data sets, because you can calculate pathway activity profiles one sample at a time, and then do the downstream analysis of joint embedding. And you mentioned that there are network level information to pathways. That is the place where I started as a network science person. But immediately you run into the problems of incomplete networks, tissue-specific networks. So it would make it more difficult. Of course, in specific contexts, our lab, I think in the second or the third publication, previous PhD students and postdocs in our lab, they took an approach of using networks, just as you mentioned, to create submodules So defined computational pathways that are not necessarily known and annotated in databases, and they've actually shown that they add to the classification accuracy and other measures. So of course, it's quite an open problem and always can be improved.